Okay, I'm going to do a quick recap of what we did in our previous lesson. Okay, it was this a class that I came in this week and I asked my first point. Okay. So you all watch this yourself, right? Okay, so now I'm going to do a quick recap. I'm going to be a narrator. Okay, let's go. Right now, you're watching the video on how to synthesize protein. Okay, this is a regular cell. We're going to go into deep dive into the cell. Inside the cell, you'll find many organelles. So the organelles we're going to focus on today is the nucleus. As we enter the nuclear pores, inside you'll find lots of DNA, condensed to form chromosomes. If you were to uncoil this chromosome, you will find that the DNA is bound around histones. We zoom in a little bit deeper and you'll see that the DNA is a double helix. You'll find that the double helix comes with many sequences of genes. Right now we're going to zoom in a little bit more, I think. Oh, no, here comes a protein. A protein comes by, and this is called RNA polymerase. The role of RNA polymerase is to unzip and unwind the gene. Following which, you will then start to bring in nucleotides to build what we call a messenger RNA. Notice that the messenger RNA is made out of A, T, C, A, U, C, and G. There's no thymine. Once the messenger RNA has been synthesized, it will then go through some form of processing. Okay? Sometimes we may snip the mRNA and recombine them. Okay, but this is out of your syllabus, just for your information. This mRNA will then be brought out of the nuclear pore into the cytoplasm. In the cytoplasm is where the mRNA will go through a process called translation. Think of this as just a recipe that needs to be translated into real food. Here, the ribosome is like a chef. And the chef will need to bring in the ingredients to create the product. You'll find that in order to create the product, things called tRNA will bring in amino acids. Okay? Each tRNA is very unique. It is able to bring in one specific amino acid. So every time ribosome is synthesizing or translating the mRNA, it will bring in tRNA that's carrying amino acids. One by one, it will piece together the amino acids into a primary sequence that will eventually fold to form a protein. Here you see the primary sequence. Once ejected from the ribosome, it will form its 3D conformation, and that's what we call a protein. Okay, so yesterday, I asked you to go home and complete some of the interactions that you did. I'm not sure if I talked about it already. Did we talk about the who and the why of transcription and translation? Okay, so we know the key enzymes involved, right? RNA polymerase. We know there's ribosome and tRNA. I told you that ribosome is the thing I want you to focus on. Right? tRNA is all extra. Yeah, Ivan, have you started recording? Okay, thanks. I then asked you to go home and do question 1. Right? What is question 1 about? Question 1 is really an image to show you the recipe book of our cells. Okay? Think of this as really the recipe book. If I say that DNA is the main recipe book, mRNA is just a photocopy of the recipe book, just a page of the recipe book. So if I give an analogy, DNA is like a cookbook with a lot of recipes. You will then synthesize mRNA. Think of mRNA as just as a copy of a page of the cookbook or recipe book. Eventually, you have your protein. The protein is like one of the dish that the chef cooks. In this case, the chef is kind of like the ribosome. Okay, so this is just a broad overview to help you understand. Some students think that DNA is converted into mRNA and converted into protein. That's not true. We cannot take a cookbook and make it into chicken rice. Right? It's two separate things. But we can use the instructions inside to make chicken rice as a guide. And so, the rest of today's lesson, we're going to try and make sense of the language, the recipe within our cells. Here, you see a table with a lot of codes. Yeah? We refer to this table as the genetic code. The genetic code is the recipe book of our cell. Best part is that it's not just a recipe book of our cell, it's a recipe book of every living organism. Every living organism works with this same recipe book. Okay, first question. What patterns do you notice when you look at this recipe book? Okay. May I get you to buzz with your neighbor on the patterns that you observe in this recipe book, and then we'll do some sharing. Okay, what patterns do you see in the genetic code? Okay, ready? What patterns do you observe in this genetic code? Eating? What patterns do you observe in the genetic code? Really subjective, everyone can have a different answer. Okay. So what patterns do you observe when you look at this genetic code? They are made of A, U, C, and G. Right? Notice that all of the codes made up A, U, C, G. Okay? There's one repeating pattern we see. Okay, anything else? Kayla? What other patterns do you observe? They all come in threes. Right? Notice that? They all come in threes. The word for such a triplet code is called codon. Okay? Say to the words. Don't need to say comes in threes or triplet nucleotide bases. Just say codon. Okay, so how do we put it in a sentence? For example, there are 4 codons that code for valine. Okay? There are 2 codons that code for asparagine. So you can use it in a sentence this way. A codon codes for something. Okay, and that something is an amino acid. Okay? So sometimes I may ask you, how many codons are present here? Yeah. So that's how we can work things out. So for example, if I were to just let's put all this on the whiteboard. C, G, C. And C, C, A. If I ask you right now, how many codons do you see? 2 codons, right? The first codon over here codes for which amino acid? Do a quick. So you practice reading the recipe book. The first codon codes for arginine. Okay, it's arginine. Yeah, first codon codes for arginine. Codon number 2 codes for proline. Okay, so that's how we will use it in a sentence. Codon. Okay, any other patterns you observe? Any other patterns you observe? Ian?
Usually it's the first and second base that determine.
Okay, notice that? The first 2 seems to be the determining factor. Right? Look at valine. The first 2, as long as it's a GU, it's going to be valine. The third one almost seems redundant, right? You can change any of the third base, and you'll still be valine. Yeah? So, sometimes, right, if you read online, they will say that the code the genetic code is degenerate. Degenerate. When you see this word and if they say the genetic code is degenerate, it just means that each amino acid may have more than one codon coding for it. Okay? Each amino acid may have more than one codon coding for it. That's just what it means. Then maybe I have a question for you. Why do you think, or what benefit might our cell have from having a degenerate genetic code? Is it a good thing or a bad thing that the third base can be changed and we still get the same amino acid? Good. Why do you think it's good? So there's more variation. Okay, in what sense? Because if I were to put in GGU and I put in GUC, it's still going to be valine that gets translated. Sorry?
So that there are more ways to code for the same amino acid.
Okay, so there are more ways to code for the same amino acid. Possible? That may be a good thing, right? Okay. Any other benefits of this? Less chances of mutation. Okay, then let's define mutation. What is mutation to you?
One of the bases change.
Okay, so well defined. A mutation is when a base changes, right? So actually, by having a degenerate code, if the third base changes, it may not necessarily change my amino acid. And that's pretty good, right? Sometimes when you all write a sentence, you may accidentally swap out a word or two, alphabet or two, right? But I can still tell what that word means, right? For example, sometimes, okay, let's just use even the word codon. Sometimes when you're writing your essay, you write codon and then after you just add one more thing, codon. Right? But as a teacher, right, I look at this, largely still the same, or I know what you mean. Yeah? So a degenerate code is kind of beneficial in this way. Right? If I swap out one code and one nucleotide, doesn't necessarily mean I change the amino acid altogether. Okay? If I want to put it, I could say in a sense it buffers for mutation. Yeah? Okay, so we've seen some methods. First one, that it is largely we use AUCG. Other than that, it comes to triplets, or what we call codons. Last but not least, you find that actually the first two are the important ones. The last one kind of buffers for mutation. Okay? At this point, some students will spot that there are some codons that don't code for anything. Can you see those codons? Yeah? You see start and stop. Yeah? Okay, think of it this way. The ribosome needs to know, it needs to be able to find the mRNA. Right? So the start codon serves as a recognition site for the ribosome. So when the ribosome spots the start codon, it sees AUG, it knows that, oh, this is something I need to translate into a protein. The ribosome also needs to know when to stop, right? It cannot just keep going on and on. So when it encounters a UAA or UAG or UGA, it knows that, hey, now's my time to stop creating a protein. Okay, so the start and stop, think of them as signals for the ribosome to start and stop. And that means to say, every mRNA will come with a start and stop. Okay? It's just like every recipe will come with an introduction and an end. Okay? It doesn't just start abruptly. Okay, I need you to remember all 64 of these codons. Okay, so if I show you, okay, don't need that. Okay, so you don't need to remember this. Okay? We will normally provide you the codons. Okay, we'll provide you the genetic code. The skill here is to be able to read it fast. Okay, don't go down to memorizing it. If you really want to remember some codons, I advise you to remember the start codon, AUG. Okay, that one doesn't change. Every mRNA will come with that. And if you notice it doesn't come with that, you have a problem. You actually cannot translate this. If you also want to, you could remember the three stop codons, UAA, UAG, UGA. Because when you see this, you know that we need to stop. Okay? So universally, all mRNA will have a start and stop. So if you want to remember something, these are the ones to remember. Don't remember the in-betweens. Okay, so I'll give you some time to jot down some notes. Okay, next question. What would the implications of the genetic code be if there are 30 amino acids instead of the naturally 20? Okay, if you look at the genetic code, the recipe book, actually there are 20 amino acids. Yeah? That means to say the proteins in your body are made from just a combination of these 20. Combination of amount and number and sequence of how they are arranged. So the question is asking you, if instead we need 30 amino acids, would using triplet codes be enough to specify for 30 amino acids? I don't know if I made sense there. Yeah, I rephrase. If our body requires to have 30 amino acids instead of 20, would this recipe book be able to accommodate 30 different amino acids? Okay. Actually, it would be possible. Okay, you look at it, the fact that every of these triplet code is unique means that if we really wanted to, we can have many different amino acids. Each triplet code technically would have coded for one unique amino acid. So actually there's an upper limit to how many amino acids we could code for. But we only need 20 in our body. So if our body does need 30, it's fine. This genetic code actually can accommodate a lot more amino acids than we actually need. For example, you look over here, asparagine, there are 2 codons, lysine also 2 codons. If we wanted to, we could have used 2 codons for every amino acid. And that will increase the number of amino acids that our genetic code can code for. Okay? But our body only needs 20 to create the proteins that we have in our body. Okay? Coming to the end. Okay, here, last one, again, for you to identify some patterns. May I get you in your pairs again to discuss what patterns you observe? Okay, because this will lead us into our final activity for this chapter. What patterns do you observe between the non-template strand, the template strand, and the mRNA, which is in yellow? Okay, a quick discussion before we dive into it. Okay, what patterns do you observe between the non-template strand, the template strand, as well as the mRNA? What patterns do you observe? Lisa? Same pattern? Everyone must think, okay, do you observe any patterns in the non-template, template, and mRNA? Okay, so Crystal notices that regardless of whether it's mRNA or non-template or template, there will always be a 3' end and a 5' end, 3' end and a 5' end, 3' end and a, my brain messed up. 5' end and a 3' end. Okay? Okay, so that's one pattern. Sakyo?
The non-template strand is similar to the mRNA.
Ah, okay.
But you just swap out the T with U.
Okay, just swap out T and the U. Okay, let's have a look. G, C, A, but swap out the T with U. Yeah? That's another pattern you can observe. So here's the thing. If I ever ask you to transcribe the template, don't waste time really complementary base pairing like how I normally do. You know what I normally do? I'll be like, okay, C means I need to put a G, C I put a G. Don't waste time. Instead, what do? Just copy over the non-template strand and then you swap the T's with the U's. Okay? Once you see this pattern, right, actually that saves you a lot of time. Okay, you got what I just said? Yeah? Okay, so if we ask you to transcribe the template, don't literally complementary base pair, waste time now. Just copy over the non-template strand. Swap out the T with you. Okay? It saves you a lot of time. Okay, with that in mind, today we're going to go into the next part of our lesson, final part. We're going to today do two things. Number one, we are going to transcribe the template. And number two, we will translate your mRNA to a protein. This is an activity I've heard that you've done before in your JH3 days. You recognize these beads? Okay, and then you have made proteins before? Okay, so in the next part of our lesson, you're going to do a quick recap to train your brain to be able to transcribe and to translate fast. Okay? Say that the more you practice, the faster you can read the cell's genetic language. Okay? So this is what we're going to do next. Everybody will work in pairs. Okay, so you look around, you have a partner to work with. Okay, work in pairs or work in triplets. Have or not? Okay, I'm going to pass around now these beads. Every pair or triplet will get one of these bags. Okay? Okay, so I know how many? 1, 2, 3, 4. Next up, every group should get one of hello, our familiar friend. Okay. 1, 2, 3, 4. Have you all smelled it? It smells damn bad. Just kidding. Why must I smell it? It's a weird thing to do. Or did I smell it? And then after I realized I was judged, then I say just kidding. Too late. Sometimes you just say something. Okay, 1, 2, 3, 4. Yeah? Okay, so if you've done this particular chapter, you should be able to go to section C right now. Section C is on mutations. Okay? Today's lesson is on mutations. Okay, I would like to draw your attention to today's activity. Okay, you're already funny, start making keychains ready, start making necklaces and okay. So you'll find that there are 6 tabs. Oh no, there are more than 6 tabs. I want you to focus on the first 6 tabs. Okay, everyone look up. There's tab 1, tab 2. Notice that the only thing that changed is group 1, group 2, group 3, group 4, and so on and so forth. Okay? I'm going to number you off, and the number that you get will represent the tab you're going to go to. Okay? Okay, if you are powerful. Wait, so you two are. Oh! Are you two a couple? Yes? Yes! Okay. So 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Chao only 1, huh? Okay. So you know your tabs? Okay. Can you go to your tabs now? Okay, go to your tabs now. Okay. Next, I would like to introduce you to the basics of mutation. Okay? There are 3 things that can happen to our genes that will result in mutation. Okay, I'm going to show you the 3 things now. Number 1. Okay, go away. Okay, can everyone look to the whiteboard? Okay, assuming this is DNA, okay, assuming this is DNA, right? Can I do a double strand? Okay, this is DNA. Imagine now on the opposite side also have C, C, G, G, T, A, G, A, and T. Okay, so it's double stranded DNA. Right, I'm going to show you 3 things that can happen to your genes, and it can be quite, it can cause a lot of problems. Okay, number 1, insertion of base pair. What does this mean? It means that actually, sometimes along the way, when you are synthesizing and replicating your DNA in S phase, this can accidentally happen. While replicating the DNA, the enzyme may accidentally insert one more nucleotide base pair than they actually should. Okay, that's insertion. You see the problem here? Okay. Normally, everything is read in triplets, right? Originally, everything was read this way. I had 3 codons to read, right? A, B, and C amino acid. When you insert something in, actually, you know what happens? We still read it in triplets. However, the new reading frame becomes this. Still A, but now we read A, C, A. And now we read T, C, T. And then A, and God knows what else is behind. Okay? So, insertion. That is the first thing that can happen. First kind of error. Let me show you the second kind of error that can happen. Rather than an insertion, sometimes there can be a deletion. Sometimes your DNA replication may mess up in the sense that they miss out one. It's also a problem. Originally, this was A, but now that you've changed and deleted one, the reading frame also changes. You get some unknown amino acid again. You get some unknown amino acid. Okay, so that's number 2, deletion. Number 3, third kind of error that can occur is substitution. Okay, as the name suggests, sometimes in replicating DNA, they may substitute something for another. This is supposed to be an A, but sometimes maybe they accidentally put a G instead of an A. Okay? Okay, so I want to let you know this right now. All of you in your various groups, I have modified your gene in one of these 3 ways. Okay? The original sequence is on tab 7. Okay? If you go to tab 7, this is the original unmutated gene. Okay, not muted. This is the original unmutated gene. Every group here has one sort of error that I put into your gene. Okay? Your task for today is this. Number 1, you will transcribe. Number 2, you will translate. And the third thing I want you to do is, can you spot which one of these errors I have included into your gene by comparing to tab number 7? Alright, that is your task for today, the mystery I need you to solve. What did I modify? Okay, you need very sharp eyes for this. Okay, so now we will have group time. Wow, 34 million views already. I'm like keeping track. Yesterday I was like, okay, sorry, distracted. Okay, so I'll give you 20 minutes. Okay, 20 minutes to transcribe, to translate, and to spot your error. Okay, your time starts now in your groups. Okay, so let you know, I don't need you to do the beat folding thing. You see all the orange images, we need to bend. Okay, I just need you to transcribe and translate. Then you go to step 3. Can you spot the error that I've introduced to your particular sequence? You spotted already, yeah? The skill to pick up today is your sharp eyes to spot errors. What error do you think I Okay, I did not spare any group. Every group has errors. Then I changed. Okay, if you think you spotted the error I introduced, let me know and I'll come by. If you think you spotted. spotted already. What's the error is? What? Okay, so what exactly is the connecting sequence? Yeah. Something changed, right, and resulted in the correct here, right? Anyone else spotted error? Spotted already? Okay. You go to Google Classroom. Go look for the page of comparison. Then you see if your shop eyes managed to catch what I mean. You spotted? So what did I do? So now you go Google Classroom. I can't do that image called Insertion. Go there and look for your error. See if you got it? Okay. You go Google Classroom. You look for an image called Okay, for those that you have applied your sharp eyes, you can check whether you are correct. Go to Google Classroom, and if you go under our class, go to comparison of mutations, you'll find this very colorful image. You can check if you are correct, right? Whether you identified correctly, whether it was an insertion, deletion, or substitution. You managed to spot the error. Okay, so what was your error? Okay, so what did I exactly do? Insertion, deletion. You have to compare to sequence 7. Compare to sequence 7. How was your sequence different from this one? In this you need sharp eyes and you will spot the error. Okay, you are good one. Good one. So you do a good one. So you say substitution. So you can go there and check. Are you all one group? Really are all correct? All correct. Is it the color reading the same? Okay, then my question is, on a genetic level, did anything change? So do you spot it? Did you spot it? So what did yours, what's the error in yours? Okay, I will go and name everything. RNA substitute. But it resulted in no change in amino acid. Okay. But yeah, so yours is one of the hardest because the amino acid is exactly the same. Even though okay, can I just have a quick check? If you are done, can you look up if you spotted your error? Right. Hey, something like the music just stopped. Okay. Yeah, so this activity was number one, a recap of what you did in JH3. But more importantly today, to train your eyes to be able to spot errors. Okay? You'll find that actually we were quite mean to some of you. Firstly, I didn't tell you that I can insert more than one or delete more than one. Yeah? Did anyone here experience a deletion of more than one base pair? Means I deleted like 3 base pairs, you know? Anyone? Have? Or some experience? Okay, so now let's run through. I'm going to teach you some of the terminologies that we use to describe mutations. Okay? Some of you have felt this impact along the way. First and foremost, okay, let's look at it. We have insertion, deletion, or substitution. First form of mutation I would like to introduce to you is this word called frame shift mutation. Okay, I don't know if this word already makes sense to you. It sounds like what it's saying. Frame shift mutation. Watch what happens. Right now, the frame of reading goes like that. This is amino acid number 1, amino acid number 2, amino acid number 3. Okay? What happens if I insert? Okay, let's say I'm going to insert now. Insert somewhere over here. Okay, this is the new fella. Inserted. Okay. And pop. Notice what happens? My frame of reading has shifted. Originally, this was my frame of reading. This was amino acid. Okay, what's ATG? Okay, this is the start codon, right? Okay, so amino acid. Notice that the frame of reading has shifted. Right after I've inserted. I don't remember where I inserted. I think I inserted over here. Okay. Originally, I read it as ATG, AGT, AGC. But by inserting one in, I now end up reading it as CAG, TAG, and then C is part of another codon. This is what we call a frame shift mutation, when your frame of reading has shifted. Anyone here experience a frame shift mutation? Okay, go ahead. Wait, raise your hand, raise your hand. Very simple, just raise your hand only. Anyone experience a frame shift mutation? Ah, so few. Okay, so I tell you which error will definitely result in a frame shift mutation? Make sense or not? Insertion and deletion confirm will result in a frame shift. Right? Okay? So if you experience an insertion or deletion, you have definitely experienced a frame shift mutation. Okay? So that's the first kind of mutation. Number two, okay, let's work this out. Originally ATG was, I think, methionine. Can someone tell me what AGT was? What AGU is? Sorry? SER. Okay, can someone tell me what AGC was? Also SER. Okay, very good. Okay, then after the frame shift mutation, can you tell me what CAG is? GLN. Okay, what is TAG? What's the new amino acid? Yeah, what's TAG? The new code. Stop. Okay, stop. Stop. Okay, so I'm going to introduce you two more kinds of mutations. Next, if the amino acid change after a frame shift mutation, we will call it a Missense mutation. It's not a mistake. Okay, it's a mistake. The amino acid has changed. This can be quite a serious problem because we know each amino acid has its properties. If we accidentally change the amino acid because the code changed, it's a problem. It is a missense mutation, it's like a mistake. Okay, next. Notice that originally it was a SER amino acid. After the frame shift, I end up getting a stop codon. Originally, my protein is supposed to be so long. Then this stop codon suddenly appear. That means now it's like this ribosome, right? Go to the toilet halfway, right? Then we shit out a little bit. Yeah? So stop codon came too early. That's nonsense. Okay, actually that's what the mutation is called. We call this form of mutation, if a stop codon suddenly appears too early, we call it a nonsense mutation. Yeah, I'm not kidding, it's really called nonsense mutation. Yes. Okay, what is missense mutation? A missense mutation is when the amino acid has changed. A nonsense mutation is when a stop codon prematurely appears. Okay, you know it's too early when actually the stop codon is supposed to appear right at the end. So if you look at the original sequence, the stop codon, by right, is there a stop codon here? I don't think I see a stop codon. UGG. I don't remember if UGG is stop codon. If I provide you an original sequence, then you should see a stop codon at the end. If the stop codon ended up appearing much earlier after I did one of these errors, that's a nonsense mutation. It is nonsense if I don't get my full product that I ordered. Yes? Okay, so now here's the question. What's the difference between a missense mutation and a frame shift mutation? Okay, so here's the thing. When you perform any one of these errors, actually, more than one type of mutation can occur. I'm going to use this in a sentence now. After I have inserted nucleotide C, the new gene has both a frame shift mutation as well as a missense mutation for the second codon. as well as a nonsense mutation for the third codon. Okay? So many types of mutations can occur from any one of these three errors at one time. Okay? So your skill, right, is to be able to spot the mutation that occurs. If I were to insert something in, can you compare to the original and tell me whether there was a frame shift, whether there's a missense, whether there's a nonsense mutation? Please stop me if I'm not clear. Anyway, okay? Yes. Okay, so Rahul asked, will it be true that every time there's a frame shift mutation, you'll definitely get a missense mutation?
Oh, unless it's only at the last codon that was added, so I have to become a stop. Then it's not. Oh, if I still change.
Okay, I would say it's really by chance. Although logic would say that if you have a frame shift, right, it's very high chance every amino acid gets changed. You may have a missense at codon 2, 3, 4 onwards. I want to show you one last form of mutation. Okay? And there was experience by Ernestine's group. They raised their hand and said that there is no error. Okay, there's no error. Okay, I want you to look at this.
Oh, it's a change in the amino acid.
Okay, you look at okay, if you look at this diagram over here that everyone looked at earlier, understand which group were you? 5. Okay, let's look at number 5. You compare number 5, if you just look at the amino acids and compare the sequence, the normal sequence, you see a change in amino acids. Exactly the same, right? Yeah? But actually understand where was the error? And which codon? 13. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, over here. Notice that number 13, originally is GAG, gap, and here is GAA. Funny, right? Okay? Okay, so GAG and then GAA. There is no change in amino acid, even though the genetics of it has changed. So this last form of mutation is called a silent mutation. We say that it's silent because on the surface nothing has changed. And so actually this form of mutation is the least serious. Yeah? Actually a lot of us have silent mutations. Secretly we have mutations here and there, but on the surface the amino acid didn't change, so we just go by with life. Okay? So the skill to pick up for this particular lesson is this. We will give you an original sequence as well as a sequence with errors. Your task is to spot the kind of mutations present as well as the error that we introduced. Okay? I find this quite fun. It's really just about your eyes shut. Yeah? Okay, I need to tell you right now that this is as easy as it goes. Because if we really want to mess with you, you know what we can do? We can insert and delete and substitute in one sequence. Possible or not? We can. We given in 1 year, right? We inserted something here, then downstream we deleted something there. You know what that means? That means the frame shift. Actually the frame shift, right? When we insert and delete. Right? So we can insert, we can delete, and we can substitute all three at one go. Yeah? So you must be very sharp and be able to compare parts. Okay? Which? Yes. Yeah. Okay, good question. If I insert 3 base pairs in, actually I'm just introducing a new amino acid. Everything after didn't really change, right? So is that considered a missense mutation? Let me think. That's a great question because we actually don't do that. At least in theory, we don't do that. Okay, but let me think.
But the amino acid didn't change. It's just the sequence change.
Yeah, I would think that the effect of it may not be so drastic. Whether you would say that the subsequent amino acids as a missense, right? I think we won't consider it. Yeah, it's as good as we inserted one whole amino acid inside. Okay, but upfront, usually what we will do is we will change up 1 or 2 so that there will be a frame shift and then it messes with you. Okay, right? More fun, right? Okay, then will you consider a frame shift mutation if I insert 3? Because in essence, right, I'm just putting in 1 amino acid, right? Is there a frame shift? To my knowledge, I think this one is not really a frame shift because when we look at a frame shift, actually everything downstream gets affected. Yeah. So to my knowledge, I think this one we can still consider it not really a frame shift because everything downstream is still kind of maintained. All we are really doing is inserting 1 amino acid. Yeah. So a frame shift, you will observe it if we insert or delete 2 or 1. Right? Then it becomes everything changes. Then your task is to be able to spot, okay, then did a missense occur, did a nonsense occur, did a silence occur. Okay? What type? There's no type. Yeah. Yeah. Okay. Oh, okay. So you're right. We actually don't have a name for that. You could just say that there was a deletion of 3 base pairs. Yeah, that's as best you can describe. Okay. Yeah. So this is a skill to pick up. Okay, I'll give you some time to take down some notes on the 4 kinds of mutation that can arise from insertion, deletion, and substitution. Okay, so you can have frame shift, you can have silent, you can have missense, you can have nonsense. Which to you is the most serious mutation? Frame shift can be quite serious, right? Anything else is quite serious? Actually, it's hard to say, right? It's really hard to say. I can only say that there's one that is the least serious, which is silent mutation. Okay, now that we learned so much about mutations, right? Actually, I just want to let all of you know that all of us are mutants. Okay, really, all of us are mutants. Because at the end of the day, what makes one allele different from another is that there is a change in the sequence, right? Almost like a substitution, right? So in other words, all of the alleles we had, there used to be only one form. Along the way, there were mutations that occurred and pop, one new allele. Yeah? And then another mutation occurred, pop, 3 alleles. Now we have 3 blood types. The original blood type of all human beings used to be O. Along the way, mutations occurred, and then we now have A, we have B. So all of us here are mutants. The fact that we look so different from each other is because at every gene, there are mutants of each other. Or we call these mutants alleles. Okay, so we are all mutants. Some mutations are worse than others. Some not so bad and can persist. Like whether you're short or tall, I mean, maybe you're not happy about it, but at the end of the day, life goes on. Yeah? Your blood type also. Maybe I don't really like A blood type, but life goes on. You're still happy and healthy. Okay? Alright, so if I were to summarize, we will classify these types of mutation as gene mutations because they occur at the gene level. Okay, it's very microscopic. The errors occur at only the gene level. The only reason why I have to bring up this form of mutation is because if we scale things up, there's another form of mutation called chromosomal mutation. Mutations have different scales of mutations. Okay? Okay, so some have asked, right, do I need to do the rest? For example, if you go to the mutation tab, there's 1, 2, 3, 4, 5, 6. You only need one of them, right? I don't need you to do the rest. So that means you will submit an incomplete SLS. Okay, then I will go and like nail everything. Okay? If we go to 8, this is the very last part of our chapter. What about chromosomal mutations? Okay? This will be your homework, chromosomal mutations. I don't have a T I'm going to give out yet. Okay? So this will be our homework. You will learn about chromosomal mutations yourself. It is not complicated, okay? Compared to what we just gone through. If you didn't find this complicated, then good for you. The next one is not going to be. Okay? I would like to give you a preview of one type of chromosomal mutation. And you understand that talk? Right? Okay. If I finish this fast, then I'll go recess earlier. Okay? Alright. So I'm going to introduce to you one form of chromosomal mutation that's related to the previous chapter. Okay? In fact, in your TE, many of you have written this down already. It is called non-disjunction. This is a form of chromosomal mutation. Okay, you watch this video now. This form of mutation occurs during meiosis. Okay, let's see what happens now.
Non-disjunction refers to the failure to separate. As this applies to meiosis, it is the failure of homologous pairs to separate during anaphase 1 of meiosis 1, or of sister chromatids to separate during anaphase 2 of meiosis 2. This first example shows non-disjunction in anaphase 1 of meiosis 1, and so you see the pink homologous pair of chromosomes failed to separate. This therefore results in half of the daughter cells containing one extra chromosome, or N+1, and the other half of the daughter cells containing one less chromosome, or N-1. In this second example, we'll show non-disjunction in anaphase 2 of meiosis 2. So you can see the pink sister chromatids on the left-hand side fail to separate into separate daughter cells. Of the 4 daughter cells produced in this example, one of them will have one extra chromosome, N+1, one will be short by one chromosome, N-1, and the other 2 will have the normal number of chromosomes, N. Now, if this non-disjunction were to occur in either meiosis 1 or meiosis 2 to chromosome number 21, this would result in one of the gametes having 2 copies of chromosome number 21. During fertilization, if one of these gametes with N+1 or 2 copies of chromosome number 21 fuses with one with the normal number of chromosomes, 1 copy of chromosome number 21, then the result is a cell with 2 N+1 chromosomes, or 3 copies of chromosome number 21, and this results in Down syndrome. Down syndrome can be identified on a karyotype by looking for 3 copies of chromosome number 21, known as trisomy 21. The karyotype will also show whether the individual is a male XY or a female XX.
Studies have shown that the ages of the parents influences the chances of whether non-disjunction takes place. As age increases, the percent risk of non-disjunction taking place also increases, up to about the age of 45.
Okay, so when we look at chromosomal mutations, we're looking at mutations on a very large scale now. Here, I have a small little insertion of just 1 nucleotide. what if you gain one extra chromosome altogether? That has its implications on a very large scale. It's like you have 3 times of a certain value, right? So there's a different scale of mutations from gene to chromosomal. But this will be your homework in self-learn. Alright, I'll just show you one process that can result in a very serious chromosomal mutation, which is if during anaphase the chromosomes don't even separate properly, and you get extra chromosomes, or you get less chromosomes than you should. There is another set of processes that you will encounter that I'd like you to read up on your own. Okay, it's not complicated. Okay, so that will be your homework, just to complete 8, and then 9, just to share about what your understanding is of how interactions fit into mutations. Right? Yes. What about? Oh, slide 7. Slide 7. Blah blah blah. In slide 7, you only have one video to watch here. Okay, you're supposed to identify the error that I included into yours and the type of mutations. Okay, so that was supposed to be what you in your test will do in class. You can watch this video for an example of a mutation that occurs. Okay, so homework for this weekend will be 8 and 9. And your corrections if you're not yet done so. Who here cannot view the correction feedback that I gave? Nicole, did I settle or did I reply you? No, not yet. Okay. So if you cannot view your feedback, you must let me know, okay? Because of the different ways in which you download and do your work, right? I don't know why the format, when I return you back feedback, some can see, some cannot see. Okay? So you must let me know if you cannot see. Alright, that's all. I'll let you go for recess now. Yeah. Oh wait, shucks. They return me the bead so that you can, see it again. Okay, so can you take out the beads, put back in the ziplock, and then I'll come around to take back my 5 peanuts. Okay, once you return to me, you may go for your recess. I will come stop the video. Thank you.